So, welcome everyone to Science Society, and of course, a special welcome to you, Anna, and Dr. Anna Schroeder. She is a postdoc at the Institute of Physiology at the University of Freiburg, and she, in her postdoc stage, had already so many grants and fellowships. She got the BBRF Young Investigator Grant, which is a pretty big deal, I think, at least here. And then she had the fellowship, Marie Curie Fellowship, and Alexander von Humboldt Foundation Fellowship, and Peter and Traudl Engelhorn Foundation, and EMBO. I mean, that's so impressive. So
Thank you.
that's
I
unbelievable.
spent so much time grant writing.
I know, but most people spend all this time and get maybe one if they're lucky. So this speaks to the impact of your research and skills you have. So
Thank you.
yeah, I mean, it's really impressive. And so different foundations think your work is really important and important to fund. So on top of a Cell publication. So congratulations.
Thank you so much.
And before we start going into your research, like, how did you discover your, passion, interest, and skill to do this type of research? Was it something you always wanted to do? Was it, did it come later on, that interest? By a class you maybe took, or a professor or teacher that you had. So yeah, if you could tell us, that would be wonderful. Thank you.
So I would say that I got interested in science quite late. So it was really sort of late in high school that I had a really great teacher for chemistry and biology that got me really interested in science in general. And then it was in college that my interest in science really grew, and I joined a biochemistry lab that was focused on neuroscience applications, and this really catalyzed my interest in the brain and convinced me that neuroscience was what I wanted to do. And in terms of the actual sort of field that I'm working in now, I really only started working on this topic during my postdoc. In the years before, all my work was actually in molecular neuroscience, so I kind of dramatically changed fields for my postdoc. And I feel like the types of questions that I'm working on now are sort of the questions that really got me interested in neuroscience in the first place. So sort of this question of how the brain at the circuit level mediates higher level cognitive functions and behavior such as memory. And so I feel very privileged to be able to work on this topic now using sort of the array of technologies that are now available for studying neural activity at the single cell level in vivo in awake behaving animals.
Yeah, that's really interesting. So it must have been quite good teachers and good school and labs that you worked in that kind of, supported you and, like, kind of woke up this 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 interest in the field. So that's really wonderful to hear that it worked out that way, because now we are lucky and that you work in neuroscience, and we expand our knowledge through you. So that's wonderful. And you alluded, how you switched the fields to do this type of work in vivo. To do this specific project, was it hard to get these grants, or did it some people come here and say, oh, I worked right away, or people didn't believe me for a long time, or it was an accident and then we wrote this grant. Was it something you planned doing for a while, or was it maybe, a discovery that kind of led to this full project? Then was it easy to get a grant? Like easy, but like did you had to write grants many times until it got funded? So yeah, if you could just give us a little bit of background about this project. Thank you.
So the grants that I started my postdoc with, I applied for 3 grants sort of at the end of my PhD to fund the start of my postdoc, and I got very lucky because all of them were funded. I think that the project was really promising from the beginning, so I think that's probably what motivated these funding organizations to sort of invest in the idea. And I think that I felt very convinced of it from the beginning, that it was something that was really worth exploring. In terms of the grants that I got later in my postdoc, there I also had to come up with sort of a new idea for my future work, and there I definitely struggled with different proposal versions to come up with the next idea. But it's sort of like this aha moment when you realize you've come up with the project that you want to focus on. And I think when you're able to convince yourself of it, it's easier to convince the funding organizations that it's something that they should also be interested in. But for the project that I'm going to talk about today, that was, that I worked on at the beginning of my postdoc, we had sort of, I think, promising reasons to believe that it was really worth pursuing. So at that time, the lab where I'm doing my postdoc now had just collected a data set that suggested that the projection that I investigated in this paper existed. And we had a reason to believe that it could be really interesting because it was a long-range inhibitory projection. And around that time, this was back in 2018, a body of literature had just been published that had shown that these types of projections in the hippocampus play a really important role in memory encoding. And so I was really inspired to figure out whether similar projections might exist in neocortex that would be involved in memory in those circuits. And I thought that the zona incerta could be a really interesting candidate for exploring that question. And luckily, the funding organizations agreed that this was worth it. Yeah.
Well, I'm happy too that they agreed. Because it's really wonderful work. So yeah, congratulations again, and I'm glad everything worked out. So, and for everyone, the paper that Anna will talk about is pinned on top of the room. You should be able to just access it, and the stage is yours, Anna. Thank you.
Thank you so much. Yeah, feel free to download the paper and go through it as I'm talking. Of course, this is going to be very casual. So as I sort of already alluded to in the introduction, sort of the core question of this project was that we were interested in understanding whether long-range inhibition in neocortical circuits might support memory. And to provide a little bit of context, so up until now, the only projections that had been studied in the context of memory encoding in neocortical circuits were long-range excitatory projections deriving from a range of different sources, such as the amygdala, the higher order thalamus. And a lot of those systems had been shown, really beautiful work has been done in those systems that show their importance for memory. But a lot of them have been shown to sort of undergo, have very common mechanisms in the sense that a lot of them, these projections target both excitatory and inhibitory cells in the cortex. They undergo sort of a common regime for memory encoding in the sense that if you compare responses to a given sensory stimulus before and after learning, in these long-range excitatory projections, a lot of them show sort of a positive stimulus response that undergoes positive potentiation, so it gets stronger. And this is all very, very interesting, but we were interested to understand whether inhibition might sort of uniquely contribute to these mechanisms. If it might undergo, if there might be sources of long-range inhibition that would sort of uniquely undergo sort of different experience-dependent plasticity that would contribute to memory. And so we decided for this project to focus on projections from a very mysterious subthalamic nucleus called the zona incerta, which in Latin literally translates to to the zone of uncertainty. And this is a name that it was given more than 100 years ago. And I would say that it's still sort of deserving of this name in the sense that it's become increasingly studied in recent years. We're starting to build a fundamental understanding of some functions of the zona incerta, but it is very much underexplored. It's a very heterogeneous nucleus in that it's composed of many different cell types, but most of these cells are GABAergic neurons that send projections really across the brain. And so we were interested to know whether it sends projections to the neocortex, and again, whether this inhibitory source might help to shape memory encoding in sensory neocortex. And if we focus on figure 1 of the paper, so the goal of this figure was mostly to sort of map and identify this circuit. So for example, in panel A, here I performed a retrograde tracing experiment from different regions of sensory neocortex. So, injecting different tracers in either visual, auditory, or somatosensory cortices of the mouse brain, and then looking in the zona incerta. And what we found was really interesting in the sense that we found that the zona incerta in adult mice actually targets all 3 of these regions, but the vast majority of neurons that we found that were labeled were actually auditory cortex projecting, indicating that for whatever reason, the zona incerta might sort of preferentially contribute to auditory behavior. And so this was one of the reasons that for the rest of the paper, we decided to focus on the auditory cortex projections. In panels C to D, this is just showing that when we inject retrograde tracers in the auditory cortex and then look in the zona incerta of mice where GABAergic neurons express an mCherry protein in the nucleus, that this reveals that most of the neurons that are sending these projections are indeed GABAergic. And you can also see this on the panel in panels F through I, where here I've injected a viral vector expressing a flexed reporter protein in the zona incerta of GAD2 cremi. So this is expressing this EYFP protein in all GABAergic cells of the zona incerta. And you can see in panel H that this is an image of the temporal neocortex, where you can see that these axons are densely innervating this area. And also that the innervation pattern that we observe is quite particular in that there's sort of a specific gradient from a sort of a mediotemporal density gradient, whereby in higher order areas of the cortex, we see denser innervation of these zona incerta axons. And we also see in panel I, you can also see it in the subset in H, that these axons are also most densely innervating layer 1, which is a really unique layer in neocortex where you have a lot of long-range afferent systems innervating from different parts of the brain. And this has sort of been coined in recent years as the possible sort of memory layer of neocortex. So we were really excited by these initial findings of the innervation pattern of this projection, and of course that it was indeed GABAergic. If you scroll down to figure 2, here we were sort of interested in profiling this circuit at a deeper level. So we wanted to look at functional connectivity of this afferent pathway with the local cortical circuit. And in panels A through G, I addressed this by performing acute slice recordings combined with optogenetics. So I expressed an optogenetic activator in GABAergic cells of the zona incerta. So this means that I was able to make acute brain slices of the auditory cortex, and then record the activity from different cell types in the cortex, and activate the innervating zona incerta afferents by applying light. And what the data here reveals is that, interestingly, this inhibitory projection actually preferentially targets inhibitory neurons of the auditory cortex. And this was independently verified. So this was, functional, looking at functional connectivity again, but we were able to verify this with an independent approach, which is shown in H through J. Here I was using a very particular anterograde tracing approach to label neurons of the auditory cortex that receive innervation from GABAergic zona incerta neurons. So I was able to label them with, again, a reporter protein here, EYFP, and then using single molecule fluorescent in situ hybridization to fluorescently label different mRNA molecules from different neocortical cell types, I was able to profile the identity of these neurons. And analogous to what we see for the functional connectivity, what I found is that almost all of the neurons that receive inputs from the zona incerta belong to either NDNF, somatostatin, or parvalbumin expressing inhibitory neuron populations. So this again shows that these afferents preferentially target inhibitory neurons and also specific inhibitory neuron classes, which are predominantly excitatory neuron targeting. And so what this means is you have an inhibitory projection from the zona incerta that preferentially targets inhibitory neurons in the auditory cortex that would normally inhibit local excitatory cells. So the net effect putatively of an activation of these zona incerta afferents would be an excitation of the local cortical circuit based on these results. If we move down to figure 3, of course, a determinant of the function of this pathway is not only the outputs, which we just looked at, but also the inputs that are integrated. And so here, what I did is I combined retrograde tracing with rabies virus tracing to map the brain-wide inputs to neurons of the zona incerta that are auditory cortex projecting. And so if you look in B, the zona incerta has been labeled with an outline, and then in this square subset, you can see a couple of neurons that are marked with white arrows that are yellow. These are the starter cells that, with this rabies virus tracing approach, we are mapping on a brain-wide level neurons that form monosynaptic connections on these neurons. And you can see that the subset of neurons of the zona incerta that target the auditory cortex is actually really sparse, but I still find this really impressive. If you look in D, this is the full data set from this experiment. Even though it's such a small subset of neurons, they really integrate inputs from throughout the brain, from different midbrain structures, thalamic nuclei, cortical areas, and regions as far away as the hindbrain and cerebellum. And obviously, this is a really rich data set, and there's a lot of things we could say about it. But one thing we noticed, and this is highlighted in panel C, is that some brain regions that are critically involved in auditory threat learning also send direct projections to these neurons in the zona incerta. So this includes, for instance, the central amygdala, the higher order auditory thalamus. And so we were really interested to understand whether this projection from the zona incerta to the auditory cortex would be implicated in this behavior. And so we directly tested this in figure 4. So here I combined a freely behaving version of auditory threat conditioning with chemogenetics. In GAD2 mice, so again, targeting only GABAergic cells of the zona incerta, I bilaterally injected a flexed version of an inhibitory dread receptor. So these are receptors that, upon binding of a specific inert metabolite, are activated, and in this case, they induce silencing of neuronal activity. And so what I did was I implanted cannulae over the auditory cortex and locally infused this metabolite there, which allowed for silencing of the axons, specifically in this region of the cortex. And in particular, if you look at B, this is the behavior that I used for this experiment. This metabolite, it's called CNO. I infused it during just prior to the session on the second day. So for those of you that might not be familiar with this behavior, it's very commonly used in neuroscience, but just to sort of give you a brief introduction. So this is a 3-day associative learning paradigm where we present the animals with 2 different auditory stimuli. These are called conditioned stimuli, or CSs, as you can see in the diagram. And in particular here, the stimuli that we use are complex auditory stimuli. And the reason that we do this is because others in my lab and also some other groups have shown that the processing of these types of complex stimuli actually depends on the auditory cortex. And so by using these particular types of sounds, we make this behavior auditory cortex dependent. And so on the very first day in habituation, we present these sounds to the animals, and they are novel stimuli, but they have no meaning for the animals. And one day later, we present these sounds again, but we now pair one of them, the CS plus, with an unconditioned stimulus, in this case, a mild foot shock. And then we leave the other CS, the CS minus, unpaired. And one day later in recall, we now present these sounds again, which have acquired an aversive significance due to this temporal pairing with the foot shock. And we can then measure the freezing behavior of the animals in response to the presentation of these stimuli to assess memory recall. And what we're looking at in C is the result of this experiment. And you can see in gray, we're looking at the control animals, and in red, we're looking at the HM40GI animals. So these are the animals that have the inhibitory dread receptor expressed in the zona incerta. And what we can see is that there's less freezing to the CSs in the animals that have the HM40GI receptor. So what we observe is that when we locally silence the afferents in cortex, by applying the CNO prior to acquisition, when we silence these afferents during acquisition, there is a memory deficit in the recall session. And interestingly, if you look in D here, we quantified a discrimination index between these 2 different CSs, the CS plus and the CS minus. And we actually don't see a difference between the controls and the experimental animals, which shows that it's not the specificity of the memory that's impacted by this manipulation, but rather the strength of the memory that these projections are impacting. And this showed that this projection from the zona incerta to the auditory cortex is important for associative threat learning. And we were really interested to understand what information is conveyed by this pathway that enables it to contribute to memory formation. And so in figure 5, you will see the behavioral paradigm that I implemented under a two-photon microscope to address this question. And in particular, this is sort of the highlight of the paper. So basically here what I did was the freely behaving threat conditioning paradigm that I just described in freely behaving animals. I implemented this completely in head fixation under a two-photon microscope. And in particular, I combined this with two-photon calcium imaging of zona incerta axons in the auditory cortex. So I expressed an axon-targeted GCAMP, which is a calcium indicator in GABAergic cells of the zona incerta, and implanted a cranial window over the auditory cortex to be able to image the axons there. And the reason that we implemented this in head fixation is made very clear from the panel in C, which shows example axons from the 3 different sessions from a given animal, just as an example. So the goal here was not only to image the same axons over days in a given animal, but really we wanted to track the activity in individual synaptic boutons at every phase of the paradigm across every trial. And so the paradigm itself is exactly the same as that which I just described in the previous figure, so we're presenting the same types of CSs with some key differences. Now that the animals are in head fixation, I'm using changes in pupil dilation as an online readout of memory. And I'm also using a tail shock now as the unconditioned stimulus. And you can see in the bottom part of figure 5b, so figure 5b shows the design of the paradigm. I also show you what are called pseudo-conditioning experiments. So this is very important. Of course, the goal here in implementing this behavior is that we want to track the responses and plasticity in these zona incerta boutons across learning. But if we observe changes in the responses, it could be because of learning, but it could also in principle be because of some other factors such as drift, representational drift of the responses that would happen anyway from repeated presentation of the stimuli. And so we implement pseudo-conditioning experiments where this is exactly the same as the threat conditioning paradigm. We present the same stimuli the same number of times, with the key difference that on the second day, the CS and the US are presented in a random unpaired fashion, such that the animals do not learn the association between these stimuli. So this serves as sort of the ideal control experiment. So when I set up this paradigm, I didn't know if the animals would really be able to learn the behavior, and luckily they are. So if you look in figure 5D, here we're looking at the average pupil changes over all sessions for the 3 different CSs. So the CS plus and CS minus, which is applicable to the threat conditioning, and then the CS1-2, which is from the pseudo-conditioned animals. And also the quantification of this data is shown in figure 5E. So we can see that on average, the threat conditioned animals, if we just focus on the CS plus and CS minus, are able to acquire a threat memory to the CS plus if you compare habituation and recall responses. And they are also able to, on average, discriminate between the CS plus and the CS minus in the recall session. However, if you look at the pseudo-conditioned animals in figure 5G, for example, here you can actually see that the pupil dilation is decreasing over sessions as the animals effectively learn that the stimuli that they're being presented with, these auditory stimuli, don't mean anything. And a really nice comparison of this data is shown in figure 5H, where we're quantifying the change in pupil dilation between recall and habituation for these 3 different stimuli. And you can see that we have sort of a threat memory strength gradient, whereby for the CS1-2 in pseudo-conditioning, the threat memory strength is low. For CS plus, it is high. And for CS minus, it's sort of intermediate. So we have this behavioral paradigm that we can use to study memory formation in head-fixed animals, looking at the activity in individual synapses. So the question, of course, is, okay, the behavior works, but what is happening in the boutons? So if you go to figure 6, here in panel A, we are looking at the responses in all of the boutons from all of the sessions, specifically in response to the presentation of the CS+. So this is, again, from all animals. And again, we were looking at the responses in individual boutons, and the only boutons that I analyzed were those that could be tracked across all sessions. So this is a paired analysis. And what we can see if we just focus on the heat map for habituation is that the presentation of the sound, which is indicated by these vertical gray lines, that this shows that some of the boutons yield either positive or negative responses to the presentation of this stimulus, as is indicated by the red or blue shading in the heat map. And this is really interesting because it shows that zona incerta axons are able to encode they're able to transmit auditory information. And this is not necessarily a given in the sense that we're looking at sort of a non-canonical pathway from the zona incerta to the auditory cortex. It's not a classic thalamic projection, but it's actually able to encode this type of information. So that was sort of the first finding. And then, of course, the question is, okay, if there are auditory responses, do we see plasticity due to learning? And the answer to this question, as you can see, is also yes. So if we look at the evolution of responses from habituation to acquisition, we see that these changes begin to manifest during acquisition, and then in recall, these responses become even stronger. And this is represented in different ways throughout this figure. So you can see a quantification of these responses in B. In C, we have some beautiful individual bouton examples. These are from 2 different boutons within the same field of view from a given mouse. And you can see how this plasticity that we observe is also observable at the level of the individual bouton. A key thing that I'll point out is that in figure 6A, all of the responses in these heat maps have been organized. They've been adjusted to be sorted based on the magnitude of the response during the sound presentation, which is why they're all moving from red at the top to blue on the bottom. But on the right side of each of these heat maps, there is an ID bar that's been provided, which shows you the rearrangement that we see over sessions for each of the boutons. So you can sort of track where they move. And this really highlights the high degree of rearrangement that we have in this system due to learning, such that the plasticity we observe occurs in all directions. So there are positive responding boutons in habituation that undergo positive potentiation in recall. There are positive responding boutons in habituation that undergo negative potentiation in recall. And the same goes for negative responding boutons in habituation. So it's really happening in all directions. And then throughout the rest of the figure, we quantified these plastic changes using a number of different approaches. This work was done in collaboration with the lab of Henning Sprekeler at the TU Berlin. And with him and a really great PhD student in his lab, Yoram, we were able to show, for instance, in panels D and E, that looking at decoding analyses showed that the ability of these axons to discriminate between the CS plus and the CS minus, it's that the system is able to do it during habituation prior to learning, and this ability gets even improved between habituation and recall. Another interesting thing to point out is in panel F, for example, and you can also see this in the heat map in A. So after the delivery of the shock stimulus, we also see a response in the boutons. So what this means is that these axons not only encode auditory information, but they also encode aversive sort of primary reinforcers such as aversive shock stimuli. Some other key things here. So for instance, if you look at panels 6I, here we're looking at the same data for the pseudo-conditioned animals, which I told you are the controls. And here you can see that these responses actually reduce over sessions. So we're seeing the opposite effect in the control animals, which really highlights that the plasticity that we see in the threat conditioned animals is due to the acquisition of memory and not because of some other possible feature of the experiment. I won't go over necessarily all of the panels here, but for instance, I can highlight panels M and N. So here we're looking at the relationship between pupil dilation as a readout of memory and the absolute Bouton response for every CS for every animal in habituation and recall. And what you can see is that this relationship is that there's only a correlation observed in panel N for the recall session, which highlights that this plasticity that we observe really encodes memory strength, which is a really interesting result. So in this figure, I have highlighted to you this unique plasticity that we observe in these zona incerta projections in the auditory cortex, and that it's highlighted by this particular bidirectional plasticity that occurs during learning. And we thought that this was super interesting, especially because you might remember that I highlighted in my introduction that that up until now, the only systems that had been studied in the context of memory encoding in neocortical circuits, in terms of top-down memory encoding, were long-range excitatory projections, which I told you are have sort of positive responses to sensory stimuli that undergo positive potentiation due to learning. So if we were to make a heat map of those results, it would have sort of this red signal that would become increasingly more red in recall. And what this means is that these negative responses that we observe have actually not been observed before. And so we thought they were really interesting and we wanted to understand them better. And this brings us to figure 7, which is the final main figure of the paper, where we decided to parse this out further by separating the groups of Boutons, the total pool of Boutons, into 2 groups. And in particular, we decided to call them the positive response during recall, or PR Boutons, or negative response during recall, or NR Boutons, because what we did was we just took the group of Boutons and separated them based on their responses in recall. But again, this is still a paired analysis such that every Bouton was then tracked from across all sessions. It's just been separated into 2 groups. And so if we focus on panels A through D for these 2 different PR and NR Bouton groups, we can see that interestingly, on average in habituation, so this is the light blue traces, on average for both of these groups, the response is actually positive to the presentation of the CS+. And we can also see that in panels C and D, that the change that is occurring between habituation and recall begins to manifest during memory acquisition itself. And we wanted to piece this out a bit further on across trials. And so this is what we're doing in panels E and F. So here you can see that we're looking at binned trial responses across habituation, acquisition, and recall for these 2 different groups. And this reveals some really striking differences, especially in the acquisition session. So if you focus on the middle panel, if we look at this for the PR Boutons on the top, we can see that across acquisition, the trace is kind of flat. So basically, there's a stabilization of these positive responses over acquisition. But then if you look between the end of acquisition and the start of recall, there's a strong potentiation of these responses. So we see that there's this stabilization feature, that then there's a contribution of memory consolidation to the responses that we see in recall. But the NR boutons, so these negative responding boutons, look completely different. So if you look in acquisition here, so in panel F, actually this transformation from positive to negative occurs during the course of the session, and it occurs very rapidly. You can see that it really transforms from a positive to a negative response over the course of the session. But here we also have a contribution of memory consolidation, in the sense that from the end of acquisition to the start of recall, there is also a potentiation of these negative responses. And just briefly, what we're looking at in G through I, this is, again, computational work from our collaborators, Henning and Yoram, where they basically showed that at the population level, it is really these negative responding boutons that drive the changes during memory acquisition itself. And so what we're, to sort of just give a brief recap, we're looking at a long-range inhibitory projection from the zona incerta to the auditory cortex, which during memory acquisition, we have a segregation of these responses into two groups that have completely different response properties, different temporal dynamics. And in particular, it is these unique negative responding boutons that have not been observed before in excitatory systems that were previously studied, that really drive the changes during online memory formation. And so, so I guess some, brief key takeaway points. Again, it's a long-range inhibitory projection that is mostly targeting local inhibitory neurons, that undergoes this really unique plasticity during learning. And so ultimately, we show here that these projections from the zona incerta definitely contribute to memory encoding in the neocortex, again, via this really unique mechanism. And I'm happy to take any questions you might have about this work.
Well, thank you so much, Anna, for guiding us through your research. This was really wonderful. And
Thank you.
Yeah, and it's a lot of work and a lot of elegant work that requires a lot of skills. So just to highlight that, and because if people are not from the field, it's sometimes, I think, not easy to understand what you need, the skills you need
Yeah.
to do very precise surgeries and so on. So, and two-photon microscopy skills and labeling. So this is wonderful work. And what I really loved is that you focused on inhibition. Back when I was a PhD student and master student and so on, I was, and later on a little bit, I also focused on inhibition. That's why, and it was not easy because not too many people cared about inhibition. So I'm really happy that that changed and that you could, precisely pinpoint the importance and involvement of inhibition in memory. So again, this is really wonderful. And I thought that was really interesting that you could correlate really these boutons with the memory formation. There's also something I observed when I studied compulsion and plasticity. Do you, in the follow-up work you may be doing right now, are you looking at what type of mechanisms are really involved in these bouton formation? Let's say what type of receptors are maybe crucial for this plasticity to happen, or maybe in the circuitry, is there any AMPA NMDA receptor ratio changes or things like that? Are you thinking about analyzing that further?
So this is a great question. For instance, one starting point for trying to think about how to move forward with this is, as you kind of indicated, where does this plasticity come from? What mediates it? And we actually did some analyses which are buried in one of the supplemental figures, where, for example, we wanted to understand, okay, in a given field of view, we have two different types of boutons, let's say these positive responders and these negative responders. Are they present on the same axons, or do they actually have, or do you have sort of mixed response types on a given axon? And what we found is that in all likelihood, you have one response type on one axon and then another response type on another axon, which suggests that probably there are two different subtypes of neurons in the zona incerta that send these projections. So it's two different groups of cells. And what I mean to highlight with that is that in all likelihood, the plasticity that we are seeing is mediated by changes in firing rate at the level of the soma of these neurons in the zona incerta, because it would be a change that's driving changes in all of the synapses on a given axon in the cortex. So there could also be a contribution of local mechanisms. So, for instance, maybe local presynaptic mechanisms mediated by G protein coupled receptors or something like that. We can't rule that out, but in all likelihood, this is mediated by changes at the level of the soma. But I do think it would be really interesting to explore if there is any sort of local mechanisms that shape this. And then I also think it would be interesting. So another thing that you kind of alluded to with asking about, for instance, like AMPA NMDA ratios, it would also be really interesting, for instance, with slice recordings to assess how these plastic changes impact local circuit activity with the postsynaptic targets that it recruits. So this is kind of a major open question from the paper, in the sense that we showed that these axons mostly target certain types of inhibitory neurons, and then of course we showed that there's this plasticity which is bidirectional, so it's not so easy to come up with an answer of how you think this is affecting the local circuit. So what it means is that we know that this is impacting the local circuit, but we actually don't have a good understanding of the effect of this plasticity on local neuronal processing in the auditory cortex. So this is a major question for future study, I would say.
Yeah, that's really interesting. I wonder if, the release probability would change, while acquiring the memory and then retaining it and recalling it over time on this. And do you? I don't know currently right now. I know that there is for different neurotransmitters, these signaling, genetic encoded signaling tools that you can basically visualize differences in neurotransmitter release. I don't know, is that something you would consider, since you do two-photon imaging and imaging in the restrained animal, I think, and you have a window in anyways, would you consider using those imaging techniques?
I've never tried imaging with anything other than GCamp. I also find these, especially because this has really, emerged in the last couple of years, that there are so many more indicators now for looking at specific responses for certain neurotransmitter types. I have not thought of a specific experiment yet where I would want to implement this, but this is definitely within the realm of possibility, and it could be really interesting also for exploring, like we were just talking about, the effect of these afferents on different local cell types, right? So that could be a really interesting tool for starting to parse that out. So I think that's a nice idea.
Yeah, we had our guest speaker group here, and they were able to distinguish um, signaling up to 3 neurons. I can send you the paper. I don't know if you read it. That's really would be probably really interesting for you since yeah, since you have the window in, and yeah, that the and they are happy to collaborate with people because they just
Mhm.
developed this, and now they would really like people to use it, and it was very impressive. So I think that would be really interesting. And
Yeah, I'll send you the paper.
did you think about kind of using disease models or stressing out the animals maybe during development or so and see if there are differences in the circuitry um, and yeah involved this pathway that is involved. Remember, let's say maybe if there was maternal stress that this pathway is stronger or weaker.
I definitely think that it would be interesting to explore other sort of aversive paradigms or anxiety-like paradigms to see sort of the involvement generally of the zona incerta, but also this projection to the auditory cortex. Um, um, uh, your first question was about. I think I got lost at
Yeah,
the first.
I think disease models or like stress. I mean, you could go on forever. There would be so many things you could do. You could do acute, like chronic stress in the animal, maternal stress, or disease models, autism, like maybe because I think that would be really interesting because um, uh, you have in humans the sensitivity to noise, especially that kind of elicits anxiety in humans. So I don't know if autism, like animals, I'm not sure about noise. I used shank mice, but my work was more related to compulsion. I'm not
Mhm.
sure if they have this noise sensitivity. I mean, you could probably, research the next 50 years. Like, but yeah, is there something that kind of made you think about using disease models?
I think that, so I guess my first starting point is that we know so little about the zona incerta that um, uh, I really want to understand sort of broadly speaking what behaviors it's implicated in, what is its connectivity, what is its function. And then I think it would be really cool to explore this further in certain disease models. What is clear from work in recent years on the zona incerta is that it's implicated in a lot of different behaviors, and that it probably processes many different types of stimuli. So it's easy to imagine that it might play a role in many things that we have not yet explored, and could also be disease relevant in ways that we haven't yet studied. One fun fact about the zona incerta that is sort of relevant for disease is that it's actually a well-established target for deep brain stimulation, and it's specifically targeted in patients that have movement disorders like Parkinson's disease. So this allows for the alleviation of motor symptoms like tremor. So that's a bit unrelated, but it shows that there's sort of therapeutic potential to studying the zona incerta, that I think if we can better understand the function of this brain structure, it could also yield like therapeutic insights that could also be studied in many different models.
Yeah, that's interesting. And I thought about that I mean, it's kind of interesting that we just go ahead and do deep brain stimulations in humans and we don't even know why it works.
Yes, exactly.
So, and then we, in hindsight, go in and actually study different brain regions involvement. But yeah, it's really interesting that this brain region is involved in so many different kind of behavioral tasks, and I think that's why maybe a lot of people scared away from it historically, because they love like brain regions like the amygdala and so on that are pretty
Mhm.
straightforward, or people at least thought that they are pretty straightforward. um, so when you look at the projections, um, does the the amount of boutons they reach change after this behavior paradigm? Did you look into that? Like, do the axons, the projections, make more connections, or is it just more in intensity, basically?
Yeah, this is a great question. So I mean, of course, it's only one animal example, but you can kind of see this in Figure 5C with the axons from this one animal example. And this is pretty representative in the sense that, interestingly, I don't really see any evidence of structural plasticity in this system. So the number of boutons or like dramatic changes in the way the axons look, I don't see this. So it's really functional plasticity that I see. So it's really that sort of the strength of the responses at individual boutons that were already there. This is mostly what we observe. Of course, there is some degree of natural rearrangement over days just because the animals are alive. So, and of course, when I'm finding this same field of view in the window over days, it doesn't always look exactly the same, but for the most part, the axons really don't change their structure.
Yeah, that is interesting, and that could give you already clues for, you could exclude already some mechanisms of, through like mediating plasticity, if that's the case. And yeah, it would be interesting to see if neurotransmitter release probability really changes, because I would assume it would if that's the case. So yeah, it would
Mhm.
be really interesting to follow your work. Um, so are you going to continue to work in this, on this projection in this brain regions, or are you planning to also switch into something else? It's just for us to have a preview of your work. Thank you.
So my plan is to keep working on the zona incerta, but I have big plans to sort of branch out. I'm still really interested in the cortical projection, but like I said, the zona incerta also has connectivity with a lot of other brain regions and is implicated in probably many different behaviors. So in my future work, I will um, really start to explore the function of a lot of these other different circuits. But I will also keep working on the behavior that I've described in this paper, because, yeah, it's a really great model for studying memory formation, and it's also a leading model for studying, the encoding of fear and anxiety in brain circuits. So yeah, so some things in the future will be related to this work, but I will also branch out. So definitely keep an eye out for my future work. Yeah.
Oh yeah, one more question. Do you plan, or would you have the availability, since you said you think that, the key lies in the soma, and they are kind of labeled. So would you be able to do like laser capture or something like that, and then send it out for RNA sequencing to maybe get the clue of what's going on in the soma? I know it's a waste, kind of a waste of animals to kind of stop probably in between paradigm steps, and then do cell capture, but it would maybe be interesting.
No, this would be something like this would be great. I would love to have a really good way to, figure out what cells in the zona incerta are sending these projections. What I think is so difficult is that I would never, at least until now, I haven't thought of a good way to be able to relate the imaging that I'm able to do of the axons in the cortex with anything that I could do afterwards in the zona incerta. Like I would never be able to know which axons derived from exactly which cells. And so, but at least there are other ways that this could be approached, and what you suggested could be one of them. Like at least looking at, for instance, I don't know, single cell changes in the zona incerta before and after learning, right? Like this might already reveal some, provide some really interesting insights. So that could help explain what we're seeing in the cortex.
Yeah, exactly. It would be a starting point. That would be really interesting. So, well, there's a lot interesting work ahead. That shows that how important and interesting your work is, because it opens up so many more experiments, questions, endeavors that can cover, I think, decades. So, unless we like have soon robots doing it all, which I don't believe. Um, yeah, again, congratulations. Thank you for taking the time out of your really busy schedule to come and present your beautiful work. And yeah, good luck. I know you don't need it because you have a wonderful record of getting a lot of major grants and awards. So, anyways, good luck for the future, and we will be excited to read what's coming out next. So, thank you.
Thanks so much for inviting me. This was really fun. I really enjoyed it.
Well, that's the also very important part, that you the speaker enjoyed it too. So that's perfect. Um, enjoy the rest of your day, and everyone, thank you for coming. Um, and yeah, follow the club if you like discussions like this. We have a little bit kind of a different, more climate related talk next, about the discovery of rivers under the Antarctic ice sheet and how they contribute to ice melting. So yeah, if you think that's interesting, join us again. And thank you, Anna. Enjoy the rest of your week, and it was really nice talking to you.
You too. Bye bye.
Bye. I'll close the room in 3, 2, 1. Bye, everyone. Thank you.